Okay, we will wait for the students to join. So it's likely that your class on professional ethics might be scheduled from tomorrow onwards. Okay, likely. I'm not sure, but it's likely. So tomorrow, I think it's from 11:15, I guess. नेहल सबको बुला लो आप.
Yes, sir, I'm calling everyone.
Vivek, you want to say something? Your voice is pretty feeble.
Sir, I was saying I heard you get what you.
Your voice is still feeble, Vivek, but that's okay. That's okay. You could put it in the chat if you want. Otherwise, it's okay. All right. Good. Okay. So what I was telling was that your am I audible now?
Yes, sir.
Okay. So it is likely that your course on professional ethics could be scheduled from tomorrow. Okay. It's most likely it's going to be held on Wednesdays from 11:15 to 12:15. Okay. We had a lot of discussions. This is not a lecture. Professional ethics class is not going to be a lecture. It is going to be more of an interactive class. Okay, and I'm sure your people are going to like it. You know, so let's hope that we will have a good time with that also. Okay. Now, one other thing that I thought I should let you people know is that let me share my screen so that you will get to know the changes that has happened, since last semester. Okay. You all knew that, this lecture being recorded, right? And typically nowadays, I cannot mark your attendance until I upload my, recording. I'm sure you may be aware, but I just thought I should also let you know. At the end of the class, we get an attendance sheet like this. Okay, so you have a record, this class being recorded and the attendance sheet. Okay, so it becomes very difficult when you're not there in the class, to mark your attendance. Okay, so this also gets uploaded. Okay, along with your Amazon attendance. Okay, so I know that in by the time you come to your fifth semester, all of you guys are much more serious. But I think some people had connectivity issues, so they were there in the class for only a few seconds, maybe less than 10 seconds, and then they had dropped out. So please make sure that you don't get into those kind of issues. Okay. Okay, let's begin our class. Um, Let me share some. So till yesterday, we talked about cloning the genes, and then after cloning, you want your primary aim for cloning is to generally express the protein, right? And when you want to express the protein, we saw that we go through a whole bunch of systems before we realize which one is going to work for us. Okay. So what is the ultimate aim of cloning? The ultimate aim of cloning is generally expression of your recombinant protein, right? What is the ultimate aim of expressing a recombinant protein? It depends, right? It depends on what kind of protein are you dealing with. If you're dealing with, say, a structural protein, like, say, actin, myosin, or any of those structural proteins, we want to understand the structure of the protein, how does it work, and all that. If it is an enzyme, we want to know where is the active site, how does the active site look like, so that if necessary, we need to design inhibitors for it, right? Or if it is a therapeutic protein, for example, insulin, growth hormones, right? You have several other enzymes that can be used as replacement therapy. Then our aim will be to make sure we purify a protein that is functional, right? You can't be purifying an non-functional insulin and expecting miraculously that it will work. It's not going to work, right? So when we purify the protein, our aim is to make sure we get a functional protein, right? And one of the issues in protein purification is that your cell, irrespective whether it's a bacterial cell, yeast cell, a baculoviral insect cell, or a mammalian cell, the cell itself has tons of protein in it, many different kinds of proteins, right? And from that, you are trying to, separate your protein, purify your protein, so that you can give it as a pure product. Obviously, imagine, recombinant insulin is being given to humans. It's 99.9% pure. Okay. In spite of the best efforts, you may still have trace amounts of contaminant, 0.01%. But will somebody take a 80% pure insulin? Probably not. In fact, when you go for these clinical trials, it mandates that your purity of the preparation should be greater than certain percentage. Okay, it again depends on many other factors, but it is generally it should be as close as possible to absolute purity. Okay, absolute purity. So that is generally the case, right? So, purifying a protein, as I said yesterday, is as much an art as much as it is a science, right? Two different people may end up purifying, insulin or something else being expressed in different platforms, and they may use a variety of techniques. They may use a variety of, chromatography techniques or, protein purification techniques, and before they end up with a purified form of protein. Okay. When it comes to academia, it probably doesn't matter whether you do it in 10-step purification or 20-step purification. You are okay. Nobody really is worried about it. But when it comes to industry, the industry is very particular that we want to do as minimal steps as possible. Why is it that? Because every purification step has a cost associated with it. And as the purification step increases, the purity of the final product, the cost of the final product also increases proportionately. Okay, for example, just imagine you're trying to, purify a protein, something like, say, our famous insulin or, growth factor. And then you realize that you have a technology that can purify it in 2 steps, and then you have a technology that is going to purify it in 10 steps. Okay. The final purity remains the same. So which one would you opt for? You would opt for a thing that does purification in 2 steps, right? Rather than having 10 steps. Okay. It saves a lot of time. It saves a lot of effort. It saves a lot of money, right? It makes a lot of sense from an economic perspective, right? Every company is out there to make at least a minimal amount of profit on everything that they do. Nobody is running a charity here. Every pharmaceutical organization, even though they may do certain things that we will see in our professional ethics class, they're not really running a charity. They need to pay their employees, they need to pay their investors, they need to pay the governments, they need to pay all kinds of people, right? And so there is a lot of business involved in it. There is a lot of cost involved in it, and so it makes a lot of sense to invest wisely and make sure you get the returns to the patient. More important, very important, that you give a good return to the patient, a good return to the people who have invested in your company, good return to the employees, right? Just imagine, you suppose you get employed by a big multinational pharmaceutical company, and they keep telling you that we can't raise your salary because we are a charity. At some point, you may leave the organization for a job that pays you maybe much better than what you're getting there, right? So to a large extent, As I said, pharma companies are not charities. They are running a business, and business is different from charity. Okay. So today's class, we will focus, I'm going to show you a short video which will talk about different steps that are involved in purification and different techniques. Okay. Let me share it. I will unshare and share it. All right. Okay. So there are two steps. One is purification, and the second step is characterizing the purified protein. Okay. So hopefully this should cover both the things. If not, we will come back and we will purify the protein, and then we'll come back and do the characterization at a later point in time. Okay. Is the screen visible?
Yes, sir. Yes, sir.
Let's say a protein or an enzyme, we need to have it isolated away from everything else, all right? When we try to understand an enzymatic reaction, for example, we say, okay, well, I'm interested in this enzyme. I'm interested in the reaction that this enzyme catalyzes. If I only have the soup of the cell, that is the cytoplasm of the cell that contains this, I not only have that one enzyme that I'm interested in, but I have several thousand other enzymes in there. So it's important for me to understand what this enzyme does, that I be able to purify this enzyme away from all those other proteins. And so understanding how to purify one protein apart from others is a very, very important consideration in biochemistry. Okay, well, there are several techniques that we use in order to do this, and I'm going to go through and sort of describe a few of the basic ones to you, and then show you some of the applications of these technologies. You can't walk into a biochemistry lab without finding a centrifuge. It's almost impossible to do that, and that's because the use of centrifugal force as a means of separating molecules on the basis of their size is a very, very valuable tool. Not surprisingly, different things can be spun down. We talk about spinning them down. That is, will they precipitate out of solution, or will they move to the bottom of the tube? The function by which they occur is a function of their size and the speed with which we spin things. So the largest things, of course, as you might imagine, spin most easily to the bottom. So if I take, and I'm interested in studying an enzyme in E. coli cells, okay? I can take a batch of E. coli cells, and I could use a fairly light centrifugation and spin, and those cells would come to the bottom of that tube, okay? Let's say I took that pellet, which we get out of that, and I'm interested in not just the cells, obviously, because I'm interested in the enzymes that's inside. I can use some techniques to bust them open. Right? I might use sonic waves to do that. I might use enzymes to do that. I might use mechanical agitation to do that. It doesn't really matter what the means I use. But when I do that, I basically open up the contents of the cell, and the insides spill out. Those insides are going to have some things in them. And in addition, I'm going to have some cell walls that are sitting there that now are empty of their contents. I could spin those down again, and if I did that, I would basically have done my first separation. I would have, on the one hand, the pellet, which would contain the very big things like those cell walls, and I would have the liquid component, which would be the cytoplasmic material that I was interested in. I could take that cytoplasmic material, I could do various centrifugations on it if I chose to, and I could separate them on the basis of the size of those complexes that are in there. Right? Now, we don't need to memorize numbers or any of that, but I do want you to understand that centrifugation allows us to do a sort of a rough separation based on size, a very rough separation. The stronger the centrifugal force, the more things I'm going to pellet, I'm going to drive to the bottom of the tube. Okay? And there's a lot of different techniques involved in centrifugation that allows me to purify things. Now, centrifugation alone will not, underline not, give me pure material. So it's used mainly as a means of what I would describe as fractionating. When we fractionate things, we break them into smaller pieces, and then we work with those pieces to do things of interest to us. Let's imagine for a moment we got two possibilities. I took these E. coli cells that I was describing to you, and I'm interested in understanding a particular protein. First question I would ask is, well, where's this protein? Is this protein in the cytoplasm, or is this protein embedded in the cell membrane? Because both of those are possible. All right? The beauty of this is, if I've fractionated it in this way, I've got one fraction that has only things in the cell membrane, and I have another fraction that has only things that's in the cytoplasm. Then I can subdivide those further, and that's some of the other things I'm going to be describing to you. Okay, so centrifugation, a very rough but powerful tool to allow us to start to separate things in the process of isolating components of cells. Another technique, yes, question? Yeah, good question. So is it actually the physical size, does density or mass play a role, and all these are variables in how things will separate? So yes, those are factors, especially as we get smaller and smaller. In some centrifugation techniques actually will work on individual proteins. And what we discover with that is that proteins that are very compact, okay, migrate through a centrifugal field very differently than those that are very open. So yes, those are all considerations, and we're not going to need to dissect those out, but yes.
So, students, did you understand the question? When you break open a cell, right, it's going to have many different kinds of proteins, and the protein sizes are also going to be very, very different. Some proteins are extremely small. Some proteins are going to be extremely large, especially those that form complexes, right? Those that dimerize, those that tetramerize, right? Some of these proteins are going to be very large, right? So your centrifugation, depending on, if you looked at the previous one, let's see. If you look at the screen, the first spin, the first centrifugation is only at 10,000 G for 20 minutes, right? The second one is 100,000 G for 1 hour, right? So depending on, what kind of proteins are there in your mixture, you could do, different gradient centrifugation so that, certain proteins will remain in the supernatant, some proteins can precipitate, okay? So let's move forward.
Start to separate things
Sir,
in the process
Yeah.
of isolating
so
components.
what was the student's question? It wasn't audible.
The student's question was that the density of these proteins are going to be different, right? When you break open a cell, the density of these proteins are going to be different. Some are going to be, extremely dense. That's what I was trying to tell you people. Dimeric proteins, tetrameric proteins, they're going to be extremely heavily dense, right? And some are going to be light. So how does this, centrifugation principle work? That's what he was trying to explain, that you do different kinds of centrifugations, and not every centrifugation is going to result in purity, but initial steps and then followed by subsequently different kinds of gradient centrifugation that is going to give you a purer preparation. Okay.
Okay, sir. Thank you.
Of cells. Another technique, yes, question? Yeah, good question. So is it actually the physical size, does density or mass play a role, and all these are variables in how things will separate? So yes, those are factors, especially as we get smaller and smaller. In some centrifugation techniques actually will work on individual proteins. And what we discover with that is that proteins that are very compact, okay, migrate through a centrifugal field very differently than those that are very open. So yes, those are all considerations, and we're not going to need to dissect those out, but yes, you're correct. They do affect things. Okay. A second technique that we would commonly use in a biochem laboratory, it's probably one you've played with in biology laboratories, either in high school or in college, and that's dialysis. Dialysis tubing is pretty cool stuff, okay? It is basically, if you've never played with one, it's basically a tube that is semi-porous. Semi-porous in the sense that it can allow water molecules and small ions, for example, to move through it, but larger things like proteins and DNA can't move through it. And in biology labs, we commonly use this as a way of illustrating the concept of concentration and osmosis. If I have a solution, for example, that has a situation here, here's my cytoplasmic mix, and let's say it's full of salt, okay, which I want to get rid of as much of the salt as I can. I would put it into a piece of dialysis tubing. The salt ions, the sodium and chloride, are pretty small. They will pass through the tube fairly readily. The larger guys, my proteins and so forth, won't pass through that tubing. And after a period of time, what I will see is that the concentration of those salt ions inside the tubing has decreased considerably as a result. And conversely, some water will actually enter that tubing. And the reason it will enter that tubing is it's trying to basically dilute out the things that won't come out. Okay, that is the proteins and so forth. So I see a pressure that arises as a result of that. Yes, Shen.
Would it be impossible to actually get rid of all the salt?
Is it impossible to get rid of all the salt that way? In theory, yes, it is, because I'm depending upon a differential concentration, and even though I get it lower and lower and lower, in theory, I could never get it completely out. You're correct. Okay, but this technique will give us a very nice, simple way of getting rid of a lot of small ions very readily. And in doing this, I have actually increased the concentration of my protein relative to the other things that are there. A technique that is a useful technique that
There
I'd
is
like to
something
describe
that's
to
very
you is that
important. Please pay
called
attention
gel
to this.
filtration, and it's also called molecular exclusion. You see molecular exclusion over here, gel filtration. We use the two terms interchangeably. I actually sort of prefer molecular exclusion, but either one is acceptable as far as I'm concerned. Now, to understand this technique, we need to understand the sort of physical nature of the separation. So to use this technique, I have to have something that's pretty cool. So I have what's called a background or matrix material, which consists of millions, or I shouldn't say millions, but thousands and thousands of tiny beads. Okay, little beads, maybe a millimeter or so in size. Big enough for your eye to see individual beads, but they're still pretty tiny. These beads have a characteristic. The beads have little tunnels through them. Little tunnels. And the little tunnels have openings that are pretty uniform in size. Now, that turns out to be important. So I've got a bead, got tunnels, and the opening of those tunnels is uniform in size. Okay? So to use this technique, what I do is I take my beads and I suspend them in a buffer. Okay? So I suspend them in a buffer, and the reason I want to use a buffer is I don't want the pH to be too high or too low. I want my protein to be stable, because if I change the pH too much, again, I'm going to denature it, unfold it, and cause some problems. So I have it in a buffer. I take that sort of buffer and contain these beads, and I sort of shake it all up and get it into a nice slurry, and then I carefully pour it into a column. And the beauty of this is that the beads, of course, can't come through the bottom. They get stuck right here. And they form a column of beads. So I've got thousands and thousands of these beads, each with little tunnels through them, each with a hole that's a set size. And yes, I can get beads with different holes of different sizes. But for any given experiment, I'm doing one size of hole for one bead, and I've got thousands and thousands of those beads. Once I have such a column, I might run my buffer through it for a little bit just to make sure that it's washed all the other junk out and so forth. And then I've got a mixture of proteins that I'm interested in separating on the basis of size. This is a technique that allows us, again, to separate on the basis of size. The exclusion part of the technique goes as follows. I've got, in this mixture of proteins, some that are very, very large, maybe 200,000 in molecular weight or greater. I've got some that are, let's say, medium size, maybe 50,000 weight or greater. And I've got some that are fairly small, maybe 5,000 molecular weight. Just as an example, I just picked those three ranges. Okay? What's going to happen with these three sets of proteins relative to these beads? Well, it turns out that the holes that I've chosen in these little beads that I've got, all right, are such that they will only let in things of a certain size. There's a size exclusion. So the great big 200,000 molecular weight proteins won't fit in the holes. They will not enter the beads at all. The 50,000 are borderline. They might be able to enter a few, but they don't really enter very effectively. And the 5,000 molecular weight proteins that I have will basically see a hole and they'll go into it, okay, just because they can. Well, if I apply these three to the top of the column, and then I let buffer sort of push everything through, what I see is as follows. The 200,000 molecular weight proteins will not enter the beads, and they will travel a very short path through the column. Okay? They just go shooting right through. They're the very first thing that comes through the column because they don't get distracted by going through all these little tunnels on the way. The 50,000 molecular weight proteins that can make it into some of those tunnels travel a slightly longer distance than the 200,000s do, and consequently follow. These would be the green ones on this display right here. Last, the 5,000 will take the longest path because they can virtually go through every tunnel that they bump into. So they take a much longer path going through the column. So this column allows me to separate then on the basis of size. The 200,000 guys coming out first, the 50,000 molecular weight guys coming out second, and the 5,000 molecular weight guys coming out third. Now, as you can imagine, when I have a mixture in cells, I have all kinds of molecular weights. So I don't just have 3 there, for example. But you get an idea about the way that we can separate on the basis of size. So molecular exclusion is a very nice way of separating these individual proteins and saying, all right, I know my protein is around 50,000 in molecular weight. I could collect this fraction from around 50,000 and then work with it further to purify. Yes?
How do you know, because your proteins obviously aren't actually yellow, green, and pink. How do you know when to, like, say, switch that you're up to the next?
Right. How do you know where they are? They're not necessarily green or red or yellow. Turns out there's a couple things that you can do. One is you can actually put molecular size markers in there that are green or yellow, which will help you. But more importantly, your question is a very good one. More importantly, I need to have a way of determining where my protein is. That means I need to know something about what my protein does. So I know my protein, for example, catalyzes a specific reaction. I could test each one of these and see where is that reaction.
So did you all understand the question that the student was asking? She said in the description here, you can see that the proteins are, different colors, right? But when you do your actual protein purification, you will not be seeing a color. This is for illustration purpose only, right? So the answer that the presenter is giving, you now have commercially that are available, you now have, suppose you are trying to purify a 50 kilodalton protein, then you can avail a protein from a commercial vendor, okay, which will be say 50 kilodalton, and it will also have a color. So you could ideally, run it along with your protein, and you can track where your protein is, whether it is going to come out of the column or not. But then you are going to have a problem because you are purifying your protein, but you're also having this protein which you added for tracking. It's going to contaminate your protein. One simple way that you can do is to collect different fractions, right? Monitor them. As the professor was saying, if your protein is an enzyme, then you can assay for enzyme activity, right? If it is a structural protein, then you could probably go for a spectrophotometric estimation, and then you will know whether your protein is coming out or not. Did I make it clear?
Yes, sir.
Reaction being catalyzed. And so I say, oh, it appears over here in this tube. All right, so now I know that this is the range where I want to collect my sample. Does that make sense? And being able to assay what my protein does is essential to purifying a protein. If I can't measure what my protein does, I have no way of purifying it. Yes, sir?
Won't some of the smallest come out with the biggest because they don't all just go into the tunnel? Some of it will go all through.
Yeah. This question is, how pure is this method? Will you get a little bit of the smallest with the largest? Again, it's kind of like the question Shannon asked about being able to get rid of all of the ions. Yeah, you will have microscopic amounts of things that's there. This is not absolute purity that we're getting. But in general, you will see the smallest come out way, way late. Okay? Yes, back there. How long does the process take? Yeah, that's a good question. It depends a little bit on the column. sometimes people really want to get as much purification as they can, and I've actually known people to pour columns that are 6 feet high, and those can take a few hours to run. If I'm running a shorter one, that might take an hour or two. So it really depends upon what I'm trying to do in terms of my separation. But there are columns.
Some real good questions are coming from students.
Yes, sir.
When you say that it's based on the size, are you talking about physical size or the weight?
Okay, so physical size and weight are related. So in general, when we talk about globular proteins, even though they have individual shapes and so forth, they, for the most part, have a given size per weight. It's not absolute, but their growth, as they get bigger in molecular weight, their physical size will actually increase as well. So it's based on their physical size, but since that's related to the molecular weight, they're sort of a one-to-one relationship. But it's not absolute. Yes?
Is this used as just primarily a rough initial process, or is this ever done sequentially where you would take a narrower range each time to evaluate a broad spectrum of sample contents?
I'm not sure I understand the question.
Like for each one of those, say you took the yellow one and it resulted from that, and then put it back through another column that had a narrower range.
Okay. Yeah, that's actually a good question also. So could I take this guy and run it through a different column that has a different size bead that might be a little bit more selective in the process? And the answer is I could do that, but there are other techniques that may be more useful to me, okay? And I'm going to show you some of those other ones. But you're right, you could do that and take it over and say now you've got a smaller bead, and so you might be getting rid of some of the other molecular weights that you don't want. But yes, you could. One of the things that you discover, just a second, Shannon, one of the things that you discover in purifying proteins is there's no one way to purify a protein. All right? You have to adapt the methods that you use to the protein itself. And you don't know before you get started what it's going to take to get that protein purified. So there may be several different techniques you'll have to use to get it, and it's going to vary from one protein to the next. Shannon?
I was going to ask, how do you know how often to change the tubes out?
How do you know how often to change the tubes out? Well, typically what people do with these is they just count drops. So I might say, okay, I'm going to get 50 drops. If the drops are coming out at a reasonably even rate, which they typically do, then people will set up fraction collectors so that every minute it'll change a tube, and that will have on average the same number of drops. Paying somebody to count, believe me, I've done this myself, paying somebody to count drops before they switch the tube is one of the most mind-numbing things that you can possibly have. So this is one of the joys of automation in biochemistry, when you've got a machine that will automatically do that for you. Okay, so that's molecular exclusion, gel filtration. Another related technique, it's related only in the sense that it uses beads, is called ion exchange chromatography. So in this method, we also use beads as we used in gel exclusion, in gel filtration. However, the beads don't have tunnels or holes in them. Instead, the beads have on their surface chemical forms that have been bonded to them that have specific charge properties. Okay? So what you see in this case is a set of beads that have on their surface ionized molecules that when they ionize give negative charge. Right? When they ionize, they give a negative charge. Now, these started out, how do I take one of these? I take my beads, and the beads start out with a counter ion. I can't get a bead that has a negative charge on it until I get it in solution and the ion comes off. So typically the counter ion might be, in this case, a sodium. I've got sodium ions out here, and they're attracted to those negative charges. So I've got sodium ions mixed with these beads, and I've got them sitting in a bottle. I take my solution, I take my buffer, and I mix it just as I did before. I pour my column just as I did before. And those sodiums are still sitting there next to these negatively charged beads. Now I've got my proteins, right? I've got my mixture of proteins. Some of my proteins will have an overall negative charge. Some of them will have an overall positive charge. Some of them will have an overall charge that's pretty close to zero. Right? So it's going to vary with the protein. How many glutamic acids does it have?
So one thing that you will realize is that when you open up a cell, each protein has its own characteristic charge. Okay, any protein that carries a lot of glutamic acid, or aspartic acid, these are going to be highly negatively charged. Okay, anything that carries glutamine, anything that carries asparagine, they're going to be highly positively charged. Okay. And then you have something like histidine. You have histidine-rich proteins, which we typically see in malaria parasite. They're going to be relatively neutral. Okay. So within the cellular milieu, you will find different kinds of proteins with all different kinds of charges. And you're basically, suppose let's just say that you are interested in purifying a protein that has a net negative charge. All you need to do is run it through a column that will bind to all the positively charged this thing proteins. So whatever flows through is going to be negatively charged. All the proteins that are rich in glutamic acid or aspartic acid or any of those amino acids. From that you can do several other techniques by which you could further purify your protein. Okay, that is how ion exchange works. Okay.
Have in it? How many lysines does it have in it? These are going to determine positive and negative charges. Well, if I have beads that are mostly negative, what will happen is the proteins that are the most positive will actually kick off those sodium ions and replace them. This is the exchange part of the name. They're exchanging those counter ions, in this case the sodium ions. So the positively charged proteins will kick off the sodium ions, and the positively charged proteins will stick, quote unquote, to that bead. What's going to happen to the negatively charged proteins? Well, guess what? They're going to come shooting right through because they don't want to interact with these beads at all. So what I've done with this technique is I've separated proteins on the basis of their charge. The most negative ones are going to come racing off. Those zero ones are probably going to follow that, and then the positives are going to follow that. And you might say, well, why do the positives even come off at all? Or how do I get the positives off? That's one of the most common things. If I want the positive ones, I've got them stuck to the beads. How do I get them off? The answer is this. Virtually every kind of interaction we talk about in this class is not a covalent interaction. These are attractive things. So if I can make something else replace those proteins, I can get the proteins to come off. All right? And it turns out if I pour a concentrated sodium chloride solution in there, there's enough sodium there, it will displace those positively charged proteins, and then I can get the positively charged proteins off. Okay? So there's an exchange. First, the protein displaces the sodium, then high concentrations of the sodium will displace the protein, and I've got what I want. So I've separated my proteins on the basis of charge. This particular phenomenon I've just described to you in general terms
Sir, the video is not audible.
Video is not audible. The questions are very, very feeble. You can hear the professor speaking?
No, sir.
No,
Not
sir.
at
positive, etc. And that actually is going to relate to another technique I'm going to talk about in a minute. But you're right, there's a whole spectrum.
So is the audio from YouTube audible?
Yes, sir. Now it is.
Yes.
Okay.
Yes,
Okay.
sir.
Of these that are there. So again, we're talking about techniques that give us basic, simple ways of separating things, but they're not absolute. I don't
I'll
get
share
only the
this
one thing
YouTube
I want
link
there.
so
I've
that
got
you
some
can go through
other
this thing.
components that are there. And there's no technique that I will tell you that is going to give you absolutely one thing. Okay, understand that. It's important. Okay. If you wonder what those anion versus cation exchange, you don't need to know these structures. I'm just showing it to you. Here's an example of something that would have negative outside. It's got a carboxyl group on there. Okay. Here's something that might have a positive thing on the outside. You can see this quaternary amine that's out there. Actually, tertiary amine that's out there. Okay. And these are commonly used. But again, don't worry about the structures of those. Okay. One of the more powerful techniques that's used in a laboratory for purifying proteins is called affinity chromatography. So like the other two techniques I just described, it also uses beads. But instead of having tunnels, or instead of having charged molecules, this technique uses specific chemicals on the exterior. So to describe this, I need to give you an idea about how I might use this technique first. Let's say I'm studying, I'm going back to my E. coli cells, and I'm very interested in a protein that I know binds to ATP. I know it binds to ATP because it uses it in a reaction that it does. So I know that this protein will bind to ATP. Okay? What I do is I take this naked bead that doesn't have anything else on it, and I treat it so that chemically it is bound to, covalently stuck to ATP. So I can covalently link ATP to a naked bead, as it were. All right? So now I've got all my beads, and they each have hundreds or thousands of ATPs stuck just out here facing the solution in the bead. All right? Well, now I take this mixture of beads that all have ATPs on them, and I pour my column with my buffer, as I did before. And now what's going to happen is proteins that bind to ATP are going to stick to this column. And proteins that don't bind to ATP aren't going to stick. Well, this is a really powerful technique, okay? A very, very powerful technique. Will I only get proteins that bind to ATP? Well, I might get a little bit of other stuff. But for the most part, I'm going to get proteins that bind to ATP. Is that only going to be one protein? Well, no, there are many proteins in a cell that will bind to ATP, but I'll have a nice collection of the ones that do, and my protein is going to be one of them. Yes, sir?
Can a bead get more than one ATP on it?
Yes, can the bead get more than one ATP on it? It can get thousands, yes. Yeah. Well, how do I get my protein? Just a second, Shannon. How do I get my protein off? I would ask you that question. How would I get my protein off of such a column? What would I have to add?
Whatever the natural target is.
ATP. I could add ATP, right? And so now my protein is going to let go of this, and it's going to grab ATP, and it's going to come off, right? That's a very cool thing, because again, remember, the protein is not covalently bound, so it's going on, going off, going on, going off. And when it comes off, a loose ATP comes in here, it binds to ATP, and now it comes off the column.
So do you understand the question? The professor is trying to purify a protein that is binding to ATP, right? So you basically, break open the cell, right? And pour the contents of the cell into this column in which ATP has been coupled to the beads, okay, microscopic beads. And anything that does not bind ATP will flow through. Anything that binds to ATP will be sitting on the column, right? The question is, how do I remove this protein now, because it is now bound to the ATP? One simplest thing will be to use ATP as such. When you have ATP, now these molecules will, instead of binding to the bead, they will bind to this ATP and they will come off, so, and that way you can purify the protein, okay? In this case, for example, he's showing you a protein which is binding to glucose. Glucose has been chemically attached to these beads. Okay, and you're trying to purify a glucose binding protein. Okay, now because these beads have glucose on them, these proteins will bind to those moieties, right? How do we elute out? By simply adding glucose. That glucose will disrupt this, okay, and these proteins can be simply be purified from these beads. Okay, so this is what is called as affinity chromatography. Okay, does that make clear? Can we move ahead?
Yes, sir.
Yes, sir.
Element doesn't stay stuck. So I add the natural ligand, in this case ATP, to the molecule. Shannon, did you have a question?
Yeah. Is it practical to functionalize your own beads, or do you usually buy them pre-functionalized?
Yeah. Is it practical to functionalize your own beads? Do you buy them pre-functionalized? You can do both. So it depends. If you have something that's a very specific molecule, you might do it yourself. Okay, good questions. All right, so affinity chromatography is really a very nice way of doing purification for specific target proteins. I want to just briefly mention one other because you frequently see it in laboratories. It's called Hplc. And Hplc stands for, and this is commonly misstated, high performance liquid chromatography. High performance liquid chromatography. A lot of people say high pressure liquid chromatography because the columns generate a lot of pressure, but in fact the correct name is high performance liquid chromatography. This is a technique for separating usually fairly small molecules, but even that's not absolute. That's been adapted somewhat over the years. The way that this technique works is by taking, and instead of using a nice glass tube that's there, these are typically poured into stainless steel tubes that have great strength. And the reason they need great strength is because these are used to at very high pressure. We don't want them to burst, for example. Okay? Well, what's the packing material? The packing material here is also beads, but the beads are microscopic. They're very, very, very tiny. Okay? So they're smaller. An individual bead would be smaller than your eye would recognize. Right? They come as powders, essentially. And these powders have on them long hydrophobic sections of molecules, like long fatty acids, for example. A commonly used one is called a C18. And what that means is that the bead has a whole bunch of 18 carbon units with hydrogens on them sticking off. Very, very hydrophobic. Okay? So now what I have, because the beads are so tiny, is I have millions of interfaces, millions of these hydrophobic molecules that the solvent is in contact with. If I pass my material through it, first of all, to get it through, it takes high pressure because these things are packed very, very densely. And they're packed densely so I can get as many of these possible things in there as I can. Well, now, instead of having charges or holes or specific affinity molecules, now I basically have a bed, all right, that is the column material. I have a bed of hydrophobic side chains. What do you suppose is going to stick to it? Well, the things that are going to interact with those hydrophobic side chains are going to be hydrophobic molecules. And the things that are not going to interact with that support are going to be hydrophilic. So now I can separate on the basis of whether something likes water or doesn't like water. The ones that will come off of a column like this first are the hydrophilics because they don't interact with those C18 groups. The ones that are going to come off last will be those that are hydrophobic, that do interact with those. The rate with which they come off is actually a function of their hydrophobicity. So we could imagine a range of things that are very hydrophilic, very hydrophobic, and things somewhere in between. What I've just described to you, and by the way, there are a couple different strategies for HPLC, but
So, just as I was telling, inside a cell, you can have these proteins that can have very different charges, right? Some could be extremely negatively charged, some could be positively charged. In general, you don't see those extremes, but you could literally, every protein will have its characteristic charge, right? Just as every protein has its characteristic charge, literally every protein also has its own hydrophobicity and hydrophilicity. Okay, so each of these techniques can be used, one after the other, when you have, so many different kinds of proteins inside a cell, and your protein, based on its unique characteristic, you could identify. Of course, nowadays it's much easier, because you now have a suite of bioinformatics tools. So all you need to know is, the moment you sequence your gene, your gene of interest, whatever you're trying to clone, it is going to tell you what the protein is going to look like, what the amino acids are, whether it is going to be overall hydrophilic or hydrophobic, whether it is going to be negatively charged or positively charged. All these tools are now in silico, you're able to predict. See, there was a time when we were doing our BSc or MSc, some of these tools were very very at the infant stage. So we didn't have any knowledge of the protein that we are trying to purify. In fact, now these days it's easy to clone a gene and purify. In those days, people didn't even know that. So they would grow an organism. For example, my first project was to clone cellulases from fungus that was eating paper. So your sole source of carbon for that fungus was paper, and by the end of like 10th day or 12th day, you will not see the paper there in the medium. Instead, it's literally gone, but you will see a thick ball of fungus. You would do a sonication, or you would add some enzyme soup so that the cells become cells lyse. Then you would take that, and then you could purify the proteins through various means. Okay, but nowadays that is not the case. So it is still the fact is, it is little different. You have cloned and purified the gene, and then now you're trying to purify your protein from a cellular milieu. Okay. In some ways it is same, in some ways it is a little different, but the challenges are different. Okay, challenges are different. But what essentially, that is why it comes back to what I was saying earlier, that protein purification is as much an art as much as it is science, because different columns are going to be used. I may prefer a C18 column, somebody else may prefer a C28 column, somebody else may prefer a shorter column. Okay, and so the question is of all the tools that are available, right? So it all depends on the final product that you want to do. If you want to do it for crystallography, your final product should have as minimal impurities as possible, because you don't want to be having contaminant proteins, right? On the other hand, if it is only for your assay in the lab for your routine this thing work, then it probably doesn't matter, because your protein and the substrate are going to be unique. So even the contaminating protein, even if it is, say, 5% or 10%, may or may not have that effect, because your protein and the substrate is going to be unique, right? So it depends on the application. On the other hand, if it is for human consumption, then obviously the performance has to be extremely different. In fact, those proteins, therapeutic proteins, use a totally different technology. The core concepts remain the same. You may use a mixture of ion exchange. You may use a mixture of gel chromatography or exclusion chromatography. Different kinds of this thing in series. Okay, but these are all done at a totally different level. I'm hoping that we can spend one lecture on purifying therapeutic proteins. Okay, but this just I wanted to show you a flavor of how protein purification gets done. Now that you people have a fairly free time, I would encourage all the students to go back and complete this, because this lecture not only talks about purification, but it also talks about characterization. This could be very, very useful for you in the long run. Okay. Any questions on the class that has happened as of now? Yes. Okay. I put the link. I have put the link in the chat box. Okay. So you could go and check that link. Okay. And if there are no questions, then either we meet tomorrow for the professional ethics class, or we will meet on Friday for continuation of this. Okay. I think I would like to wrap it up with this thing, and then we will probably start looking at therapeutic cloning, basically the cloning of animals and all that from our this thing. Okay, next week only. All right.
Thank you, sir.
Okay, take care. Bye.